Hello, everybody. I'm Donal Murphy, Marketing Programs Manager at PacBio, and I want to welcome everybody to the PacBio webinar series. Today's topic focuses on our new application-free targeted sequencing application powered by CRISPR-Cas9, allowing you to enrich and sequence even previously inaccessible regions of the genome. It's my pleasure to introduce two of my PacBio colleagues, Jenny Ekholm, Senior Scientist, and Paul Katuri, Director of Product Management, who will be presenting on the topic. We have a lot of great material to cover today, and the presentation portion of the webinar will be followed by a questions and answers session. So you're welcome to submit your questions at any point during the webinar by typing them in the area provided on your attendee dashboard. We will be recording this webinar and making it available for download in the next few days, so please keep an eye out for a follow-up email with a link to the recording. So with that, we'll get started, and welcome, Paul.
Thanks, Donald. Jenny and I are excited to talk to you today about the NoAmp technology. We're just going to, for the agenda for today, is going to be a brief overview of smart sequencing for those of you who are not familiar with PacBio sequencing. And then Jenny will take over and provide the bulk of the presentation, talking about NoAmp targeted sequencing, unlocking repeat expansion disorders with this technique, and then talking about the overall capabilities of this technique. And I'll conclude by a quick product overview and help moderate the Q&A sessions. So just looking at a big picture, what can single molecule real time, which is smart sequencing, do for you? We characterize our smart sequencing by having long reads. And by long reads, we mean tens of kilobases of read length. And I'll show some examples where our read lengths on average are well over 100,000 base pairs, maxing out at 250 to 350,000 base pairs. Now, not all these sequences these inserts have to be that long. We can sequence inserts, PCR products, say from 500 base pairs all the way up to inserts well over 50 kb and higher. One of the main features of our long reads is high accuracy. And we achieve that high accuracy because we're free of systematic errors. And we can easily achieve QV50 through our consensus sequencing. And what we do, and I'll show some examples of this in the next slide, is we actually sequence every individual molecule that's put into one of our very small wells. We can sequence DNA or RNA, and these long reads, again, can be very high accurate. They can easily achieve Q20 and even Q30 read accuracy. Because we don't amplify our genomic DNA, and our polymerase does not have any sequencing bias, we have no DNA amplification bias. So we can sequence high GC and high AT regions and sequence highly complex regions without any bias. And lastly, because we are sequencing in real time, we're actually monitoring the polymerase adding bases in real time, just like a movie. We can actually detect when the polymerase comes upon a modified base because it slows down a little bit trying to figure out the correct base and adds it in. That slowing down, that slight pause, allows us to characterize epigenetic marks. What we have here is we have a series of wonderful reviews and great demonstrations and proofs of our technology, and we have thousands of publications of our technology. And sort of a high-level view of our workflow is we can take in a huge variety of samples. Here it says from viruses to vertebrates. We isolate the genomic DNA or RNA, and one of the key things for our template prep or our sample prep is be able to ligate on these hairpin adapters. And what that means is, if you can see the insert that we show here in yellow or orangey and blue, we create circular molecules. We ligate these hairpin adapters on each end to generate what we call small barrel libraries. Then very simply, anneal our sequencing primer and bind our sequencing polymerase and load the samples onto the instruments. We add sequencing chemistry and smart cells and close up the door, and away we go. And the magic really happens in the very small wells, which we call zero mode waveguides. Our goal is to get one polymerase per well, and it's shown here in the diagram here. And these circular molecules, the smart ball libraries, they drift towards the bottom and they tether themselves. The polymerase is tethered to the bottom of the well. And then the polymerase in real time goes round and round the template, and creating multiple copies of that single template. And that's shown in this next slide. Here, you can see here's our polymerase, and this is a diagram of going around this molecule. And once you stretch out the sequence, you can see that the two strands of the DNA will line up. And we have some random errors, but these are not systematic errors. And these random errors will ultimately provide what we call a CCS read, which stands for Circular Consensus Sequencing Mode. And these are very highly accurate. We'll show an example of that in the next slide. We currently have two main systems on the market. They're here in yellow's example data from our Sequel system. And then earlier this year, we've launched the Sequel 2 system. And the main difference is throughput. The Sequel system has 1 million ZMWs, or 1 million wells, and the Sequel 2 system has increased the throughput 8-fold, and it has 8 million ZMWs. And this is a distribution of read lengths you can see, and as I mentioned before previously, the read length here can approach well over 200,000, as high as almost 300,000 base pairs. And this typically means that over half of the data is greater than 190,000 base pairs. The longest reads are this 200 to 300 KB, and data output varies between 50 gigs to 160 gigs, and which corresponds to the Sequel and the Sequel 2 systems. So what does it mean when you kind of combine this amount of data and these long read lengths and the circular consensus? It means that going around and around for the molecule, an average of 10 times, this is the number of the coverage for that individual molecule, we can actually achieve a QV30 from molecules that are 10 to 20,000 base pairs long. And that's for 10 passes. You can, with Sequel, you can achieve QV30 with 10 passes, and Sequel 2 is even a little more accurate. You can actually achieve QV30 with only 8 passes. So right now, with that background, I'm happy to turn it over to Jenny Ekholm, and she will walk us through the.
All right, so let's switch gears and talk about the NoAmp targeted sequencing method. So as the name indicates, this method is completely amplification free. And we can achieve this by using the CRISPR-Cas9 system that for most of you, you're familiar with as a gene editing tool. But what we have done is we have re-engineered the tool. So instead of putting a sequence into the genome, we will actually pull out the region of interest. And by combining this with smart sequencing, and you already heard all the benefits from Paul, the long reads, the high accuracy and uniform coverage, we can now sequence regions that's previously been unsequenceable. So all in all, this method is really great for targeting regions that previously been inaccessible to both PCR and sequencing. Okay, so let's switch over. I'm going to give you a visual overview of the method. I'm going to talk about the method a little bit more in detail in the next slide. But just briefly, you will start with your genomic sample and extract the DNA. And after that, you will block both the 3' and the 5' ends of your genomic DNA. And you will do this just so that it's not accessible for ligation in the future steps of the protocol. Now, after this, you will go into the CRISPR-Cas9 digestion step. And in order for the CRISPR-Cas9 to pull out the region of interest, what you need to do is design a guide RNA at each end of your region of interest. Then the Cas9 will come in and recognize the guide RNA and do a double stranded cleavage. And this is basically what you will get. You have a DNA fragment that contains your region of interest with free ends. And this is important since all the other ends now are blocked. So that means when we introduce the sequencing adapters, the hairpin adapters that Paul was talking about, they can only bind to fragments that have free ends. So it will only be available to the ones that have your region of interest. Next, we do a cleaning step, and then we take it in for sequencing. And the sequencing can be done on both instruments. So Paul showed you both the Sequel 1 and Sequel 2. And after you've done the sequencing, you will take the output data and bring it into our Smart Link software. And after that, you can take that output and input it into different visualization tools that we'll talk about a little bit later. So all in all, again, the NOAMP protocol is compatible with both of our sequencing systems. It's a two-day protocol, roughly. You can choose to multiplex several target regions if that is what your study design needs, or you can also choose to multiplex several samples. The input genomic material is 5 to 10 micrograms per smart cell. Now, depending on how much you multiplex, that can be then subdivided up between the samples. So let's say, for example, you want to multiplex 10 samples. That means you have 1 microgram per sample input material. The insert size that we have mostly worked with in-house is 4 to 6 kb. That said, we've done some tests for larger inserts like 16 kb successfully. In the protocol, you will see we have validated 4 different targets. And when I say validated, that means we have all the guide RNAs and everything already outlined in the protocol. And this is for the Huntington's disease locus, for the Fragile X disease locus, for spinocerebellar ataxia disease locus, and from the ALS disease locus C9 or 72. All right, so let's jump into the protocol in a little bit more detail. As you can see, these are the main steps that I've already mentioned. So you do the 3' and 5' blocking of the DNA ends. Then you do the CRISPR-Cas9 digestion. And then you do the adapter ligation, and then do the library cleaning, which is done with exo combined with the trypsin treatments, and then the sequencing. So in between, there are some steps of ampere purification, but other than that, it's very straightforward. You have 2 optional stopping points in the protocol. One is before you take it into the cleaning step, and one is before you take it into the sequencing part. Now, you're looking at a total of incubation time a little bit over 10 hours. And in terms of hands-on time, it's a little bit below 4 hours. So this can be stretched over 2 days. How you divide up the days is dependent on which stopping point you want to choose. So once you have the sequencing data, you take it into the data analysis. So again, the first part is to make this really highly accurate reads. So going around the molecules at least 3 times, and that will give you a very high consensus accuracy. And this is all done on our Smart Link analysis system. This is very easy to use. This software is a GUI that you can put on your laptop, and there's all drop-down menus. So it's very straightforward to do. Now then you can take the output files and you can do different things. So one is we have a command line tool for you to summarize the results. So you can just have a quick look what it looked like, the regions you captured, read lengths, and repeat lengths if that's what you're looking at. You can also visualize the data. We have several different options here. So one is IGV, or Integrated Genome Viewer from Broad Institute. And here's an example shown here. Then we have a few other tools available to you through command line. And I'm going to talk a little bit more about these later on in the presentation, but these are specific for anyone that's looking at repeat expansions. All right. So one of the genomic regions that generally is quite difficult to amplify and sequence are repeat expansions. And for the same reasons, these are also prone to cause disease. And these disorders are called repeat expansion disorders. And there's over 20 such disorders that are known, and these are all signified by having an expandable repetitive units, and they can really reach from below a kb and for some even hundreds of kbs. And the way this works is if it expands beyond a certain threshold, it actually causes the disease. Now, there's some shared themes between these disorders. So one is interruption sequences. So these are repeat elements that are found within the repetitive region. And so far from what's been seen in the research, they act as phenotype modifiers. And they can all, they're a little different for different disorders. And another shared theme is also repeat length heterozygosity or mosaicism that is seen for the expanded allele. And in addition, for some of the disorders, there is also believed to be a epigenetic factor that play a role. So what you see here is the chart of the most common repeat expansion disorders here on the left. The boxes indicate the range of the expansion for that repeat expansion disorder. And for the blue part of the boxes, that indicates the expansion size that's seen in the general population, and it does not cause disease. But once it expands beyond that, so you can see the gray box, it's called a pre-mutation state. So this is a state where the individual can show some less severe signs of the disease phenotype or have a completely different phenotype to what is seen then for the full expansion. So once it proceeds to the full expansion, the individual will get the repeat expansion disorder. So what you can conclude from this slide is that most of them really falls between below 1 kb and 3 kb. We do have some outliers that in some instances it's been reported that their expansion sizes can be well over even 60 kb. So for most of these disorders, the genes were identified already decades ago. But because the researchers could not really find a technology that they could get the sequence information of these repeat expansions, they've been having a real hard difficulty to really understand the full biological disease mechanism still to date. So to progress research, they really need the base level resolution into these regions, and that will then allow them to get an accurate repeat count, which will give them a really much better understanding where is the threshold for the normal repeat length for the general population, pre-mutation, and the full mutation. And they also need to be able to identify all these interruption sequences. For some disorders, there's some evidence already of it, but for many, this still remains a question how they affect that disorder. And then lastly, but not least, they also want to look at mosaicism of the expanded allele. There's some indication that this might have an effect on the phenotype severity in some of the disorders. So all in all, if they have all this information, it is going to be much easier for them to better understand the underlying disease components and in the future then provide better diagnosis and treatments for the patients. So let's look about on the traditional methods that usually are used for looking at repeat expansion disorders. So really it is PCR and Saturn blot that are mainly used. PCR usually is used for anything kind of around 600 base pairs, where Saturn blot is then used for the larger expansions. With PCR, although you can get a repeat count, it's still, since it is amplification, it's still, there's still a chance for PCR bias and copying errors. Now, if you want to look at interruption sequences or somatic mosaicism, you can. However, both of these would require additional experiments. So all in all, you would need to have 3 different experiments to achieve that information. And for Saturn blot, you won't really be looking at the actual repeat count to the base pair. It's more of an average where the repeat size lands. And if you want to look at mosaicism again, you would have to do an additional experiment. Now, with the NOAP method, you can really get all of these points or all these data in one experiment. So you get the base level resolution, which you don't get with the PCR and Saturn lots. You get the actual sequence, and that's going to allow you to do an accurate repeat count and then identify the interruption sequences. And of course, since we're using the CRISPR-Cas9 enrichment method, there is no PCR bias or copying errors. And then you can also, because we're looking at single molecule, highly accurate single molecules, you can also elucidate the mosaicism of the expanded allele. So all in all, you can do everything holistically in one experiment. We have had several collaborators already that have demonstrated the utility of the NOAP method in diverse range of repeat expansions. And here is just listed some of the peer-reviewed publications that have been published so far to date. Okay, so let's talk a little bit more about the capabilities and expectations of the method. So again, as I mentioned earlier, the insert size that we have mostly been working with in-house has been between 4 and 6 kb. But we have tested also larger inserts. You can choose to multiplex both targets and samples, or you can choose to just do one target and sample. So it really does give you that flexibility. Now, the input genomic DNA needed is really dependent on your multiplexing level. So again, it's 5 to 10 micrograms per SMRT cell, and you can then subdivide it up if you are multiplexing samples. So what you're really looking at in terms of the on-target yield, so you're going to get hundreds of CCS reads. So these again are the highly accurate reads where we have consensus sequencing with a minimum of 3 passes around the SMRT bell per 1 to 2 micrograms of sample. Now, the on-target rate, so this is when we're looking at the sequencing chips, so what we refer to as the SMRT cell. So for, you're really looking at all these wells that are being sequenced, so 40 to 60% of these wells, from the wells that have sequencing data in them, do represent sequence from the region of interest. And this really translates into an enrichment factor of tens of thousands to even hundreds of thousands fold. Now, this method is target agnostic. And what I mean with that is that you can really use this for any part of the genome as long as it's within the reach of the CRISPR-Cas9 system. That said, if you are looking at repeat expansion disorders, so what you really can expect to see is a base level resolution into your region, so you get the sequence. And that will help you to do the accurate repeat counts and then identify interruption sequences and characterize the mosaicism of the expanded allele. Now, how much coverage you need really depends on your study design. So if you want to look at mosaicism and really want to capture the entire range of the repeat length sizes, we recommend to use at least 50 molecules per allele, so 100 molecules total per sample per target. Now, if you're not interested in that and you just want to have an average repeat length call, you can get away with approximately half of that. Okay, so let's take a look at an actual sequencing run using the NOAP method. So this run has been done on the SQL system. And what we did is we had 4 targets that we multiplexed. We had the HTT, FMR1, Taxane 10, and C9 or 72. We had 5 samples. So 1 control DNA and 4 CORIELL samples that had the disease expansion. So you had 3 samples with HTT expansions, and these were all of different size range. And then we had 1 with FMR1 expansion that was quite large. We had used input genomic DNA of 1 microgram, so 5 micrograms total for the entire run. And what we saw was on-target ranges, reads that range from 30% to 65%. And this translates into enrichment factors of almost 24,000 to 237,000 fold. So if you look at this run, again, if you want to look at mosaicism, it's 50 molecules per allele. So you have 2 alleles, so that's 100 molecules total. So for most of these samples, you could very comfortably call the mosaicism. Now you can see that there's one outlier here. And this is because the sample is not as of good quality as the other ones. So what we see is if you have fragmented DNA, then you're likely to not get as high of a yield when it comes on target yield as for samples with good quality DNA. Now, if you want to increase the number of targets, well, first of all, you want to use high quality DNA. That will help much with that. But you can also increase the input DNA amount. Now let's look at the second run here that is done on our Sequel II system. Again, it's the same targets, same samples, but here we use 2 micrograms per sample, so 10 micrograms total. And the on-target rates were very similar as for this previous run. But what you can see from this one, you definitely have much higher on-yield target counts for each of the samples. Again, this one is a little lower because of the poor quality, DNA quality. But what you can see, you can really comfortably call the mosaicism for each one of them. In fact, you could actually increase the multiplexing level by quite a bit and still get the results. So really, when you think about the question is, how many samples can I multiplex? So that really comes down to your study design. What is the aim of the study? Do you want to look at mosaicism, or do you just want your average call length? So then you know how much you need, how much coverage you need, and then you can calculate that, how many you can then comfortably multiplex and still get the results that you're looking for. Now, in terms of targets, we have a multiplex 4 here. We saw no decrease in yield by multiplexing targets. We have multiplexed up to 6 in-house, and we still haven't seen any drop in yield due to the multiplexing. So that just is an indication that you can go much higher if that is something that your study design requires. Okay, so let's look at how the genotype calls compare between the traditional methods and the NOAMP method. So this is what we call motif count plots, and this is available to you as part of the visualization tools that we offer for the NOAMP. And what this one does is it allows you to look at the repeat count and characterize the mosaicism. So what you see here is the number of CCS reads, so the highly accurate reads that you have. And then here is the number of repeat counts. So this is not bases, it's the repeat count. So in this repeat count, you have 3 bases. So this is basically where it peaks. Here you can see for the normal allele, it peaks at 22 repeats. And for the expanded allele, it peaks at 50. And you can see that there's a perfect consensus between the CORIELL call and the NOAMP genotype. Now, similarly for the 4 other samples that we ran in this experiment, you can see that they're quite similar, the calls where we have the CORIELL information, genotyping information. However, once they get a little bigger, the PCR tends to start breaking down, and there might be some small discrepancies here. And then for the really big expansions, as here, where you have doing basically Saturn blot, you just have more of a range. So all in all, you can really see how valuable the NOAMP method is in giving you a much more detailed into your results. So you can really see where the repeat length sizes land, and it's very useful for looking at mosaicism and the spread, and it gives you much more details than, for example, with the other methods. So one thing you can also do with the NOAMP method is look at the CRISPR-Cas9 off-target effect. So this is something that really comes down to how specific your guide RNA design is. So normally we have what we call a reference guide, which contains step-by-step that you need to go through in order to design guide RNAs for the experiment. So normally, if you follow those instructions, we actually see very little off-target effects. So for example, for this run, so you can see the coverage level here And here are the chromosomes. You see the HTT is really highly covered. Same thing with the ALS, C9orf72. And same thing for Cas9 and the FRMR1 targets. Whereas you see there's a little bit off target here, but the coverage levels are really not that high. So it really does give you a pretty good indication that the guide RNAs used in this experiment are very specific. So this is another tool that is part of our NOAMP visualization toolkits. Again, this is a command line tool, and what you can use, what you get is basically what we refer to as waterfall plots. So what they really allow you to do is visualize the repeat structure, and then to that you can of course identify interruption sequences. So the first example here is Huntington's disease, and here you have the normal allele, and here you have the expanded allele. And you can see the normal allele has several different interruption sequences present, whereas the expanded allele, in the expanded allele, these are mostly absent. The same thing for the Fragile X example. You have here the AGG interruptions. So you find 2 in the normal allele, whereas you just find 1 in the expanded allele. So this is very useful for just mapping out interruption sequences in your samples. Now, there's another visualization tool, as I had mentioned earlier, available to you, which is the IGV viewer, or the Integrated Genome Viewer. This is freely available from the Broad Institute. And what makes this tool nice is that you can look at the entire region you captured. So usually, as you saw from the slides in the beginning, you capture your region of interest. So usually that has an expansion or something else, but then you have some buffer sequence on each of the ends. So this one really shows that entire region that you have sequenced. And that can be quite useful because sometimes you might find really interesting variants close to your expansion site or your region of interest site that actually might be important for your research to know. And the other nice thing about this is you can really zoom out and zoom in how much you want. So you can really go into a base level resolution here, and you can really look at your sequencing data in very great depth. Okay, with that, I will hand this over back to Paul, who will be talking more about the product.
Thanks, Jenny. That's a really great overview and the capabilities of the NOAMP overall technique. And as I get back into presentation mode, here we go. So just a few more slides here to describe the product and how you can start actually using this technique in your laboratory. One thing before we talk a little more about that, I do want to remind you, we talk about high quality, high molecular weight genomic DNA, and really what that means is to be able to achieve the 1 or 2 micrograms per sample. We really like to see that the distribution, the mode of that distribution being greater than 50 kb. And we've worked with multiple partners of ours to validate genomic extraction techniques. If you're unfamiliar with those, we can reach out to tech support. So our target is 5 to 10 micrograms per cell, and as Jenny's shown nicely, you can multiplex different targets or different samples. So what that means is we can routinely and robustly get the 50 to 100 CCS reads per target per sample with only 1 to 2 micrograms of genomic DNA. And probably if you're starting off, we would recommend you start off with 2 micrograms, and then we will also provide you with what guide the sequences or guides the sequences of which guides we validated, and you can probably wise to add one of those in as a starting point as you look at designing targets for guides for your particular target. Okay, quantitating genomic DNA is important, and then we have a reference guide which we'll talk about this, but it is very easy to multiplex multiple targets, and we've gone up to, we showed here 4, and we don't see any degradation in sequence as Jenny showed. The guides are fairly specific, so we don't have an upper limit for the number of targets you can assess right now per sample. In terms of the only new PacBio product that, other than our standard workflows and smart cells and sequencing kits and binding kits, is something which we call the NoAmp accessory kit. Now, I'll show the recommendations of third-party reagents on the next slide, but this kit it contains our sequencing primer, a buffer, and some annealing buffer, which you will use to anneal the hairpin of the barcoded adapters that we showed previously. So what do we have in terms of documentation? We have this fully supported protocol from a reference guide on how to design a new guide, and we typically recommend third parties. We found them to be the most robust for designing new guides. For PacBio specific library prep or sample prep protocols, we have a procedure and checklist. All the analysis tools that Jenny has nicely described to you. There is an overview document, an analysis protocol document, which walks you through what is done within SmartLink, which is our GUI-based tool, all the way through to some command line tools that are very easy to use to be able to visualize some of these results. Standard for every product release is a product insert, and those of you who are interested in porting this protocol over to Sequel 2, we're in the process of launching a Sequel 2 protocol in the upcoming very short time period. So I mentioned third-party reagents are required, and in the procedure and checklist, what we call the wet lab protocol, there's a list of third-party reagents. Everything is be able to source worldwide and globally. We have a recommended CRISPR-Cas9 RNA oligonucleotide sequences from IDT, which is our typical go-to vendor for these. If we have validated a guide for some of these repeat expansions, you'll see the sequence here. And these are pretty low cost. In the US, the list price for these guides are about $100. Oh, sorry. And I forgot to mention, we also, since you're going to be multiplexing multiple samples to reduce the cost as much as possible, and I'll talk about what these costs are on the next slide, we provide the sequences for the barcode adapters, which are, you need one barcode adapter per sample, not per target, but per sample. For every sample, you want to assign a different barcode. And then on our website, we have an oligo order sheet, which you can simply download. Choose the number of barcode adapters that you want to order. So if you want to do 5, 10, or 20 plex, and then order those through your local oligonucleotide supplier. So what does it cost? So if you're assuming we're starting with 1 to 5 micrograms of input, these are typical recommendations. And as Jenny alluded to, if you have some poor quality genomic DNA, you might want to up this for 5 or maybe a little bit higher. And that might mean you do 2 of the sample prep reactions instead of just 1, or just doubling the volume. But assuming you're in this 1 to 5 microgram range, this is all in consumables price, both third-party reagents and PacBio sequencing reagents. If you do about a 5-plex, 5 different samples, and then you can do multiple targets, but 5 samples, the US list price is about $220. If you double that to a 10-plex, it's about $130 per sample. And if you're doing multiple targets per sample, then I mentioned these guides are about $100 for each one of them. You need 2 of them. You disconnect these because you get such a large amount for the minimum order. This price can be amortized over well over 200 samples, and that drops the overall price down to about $20 to $40, including all sequencing reagents and upfront analysis. But do want to recognize that there is an initial purchase, and want to provide that transparency because you need to buy one of everything that I've listed on the previous slide. You will need to have an initial outlay. These are, again, approximate US list prices through IDT. There will be initial cost of $3,500, and that the adapters from IDT represent about 37%. We've also worked with Sigma oligos, their gold standard synthesis, and because their Sigma is a little bit larger synthesis size, that price is $8,000. And you can see in both cases, a lot of the purchase price is wrapped up into the oligos, which once you've purchased it once can be amortized over hundreds of samples. But to kind of start wrapping things up, Jenny and I have provided a nice overview of smart sequencing, We talk about long reads, tens of thousands of base pairs, highly accurate reads by generating consensus sequences. We have uniform coverage. We have no bias. Some of the examples are almost 100% GC, especially with the FMR1 loci. So we believe that we can now achieve sequencing the unsequenceable low complexity regions. We have single molecule resolution, which allows us to detect both sequence interruptions, which can't be achieved with PCR or Southern blot, and also be able to start characterizing mosaicism. This technique in general is amplification free and with no PCR biases or errors. And we have demonstrated this capability with what we believe is a highly relevant area of science, as well as a difficult technical area of sequencing and enrichment through the demonstration of these sequencing and analysis of these repeat expansion disorders. And we believe we can achieve a holistic approach and capture all the information that's needed in just one single experiment by accurately counting the repeats, identifying interruptions, and characterizing mosaicism. With that, Jenny and I would like to start thinking about or answer any questions, and just a generic place where you can look up. There's a lot of great information on our website, pacbio.com and NoAmp. If you have any questions that we can't answer quickly today, you can please reach out to a PacBio scientist through this URL. And at this point, I'm going to open this up to questions. And Jenny and I will be looking forward to trying to answer the most questions as we can. Okay, I think in general, Jenny, are you with me?
I am, yes.
Thanks, Jenny. Jenny, in general, there are lots of questions, which is great. In general, there's a lot of questions around the size of the insert that we've looked at, and how big would you expect we could go? So can you talk a little bit about that?
Yeah, so basically in-house or internally, we have mostly focused on 46 kb sizes, and that's just mainly because we've been looking at repeat expansion disorders, and we can confidently capture most of those with that insert size. With an expansion, of course, for some of these, that's the insert size, but then with the expansion, they can be much longer. Of course, for some of the larger expansions. We have tried, tested some up to 16 kb. That doesn't mean that that's the end all. We just haven't really tested it too much to see where it breaks down. But at least up to 16 kb, we have seen, we tested so far. So what you really need to think about is then the CCS reads. So you want to have this highly accurate reads, especially if you want to look at mosaicism, and that will be your biggest limits on how far you can go if that's what you want to do. You can also, of course, acquire consensus through just several different molecules if you're not really interested in the individual molecules per se and the mosaicism. So then you can probably go much higher in the insert size, but we really haven't tested what the proof is on this one.
Yeah, that's a great answer, Jenny. So, and I think in general, you know, our target is to provide these highly accurate CCS reads of a few passes, 4 or more in general. So, as a core technology, we're if you look at human resequencing, we're pushing the insert sizes right now, 15 kb, 20 kb. So we're launching this new capability with NoAmp, and we've started in this 4 to 6 kb and plus region, as Jenny has alluded to. But we believe we can accurately, easily get up to this 15 to 20 kb region, and still produce reads of 220 or higher. Was that fair to say, Jenny?
Yeah, that's good.
Yeah, fantastic. Yeah, okay. So let's see. There's a quick question around costs compared to PCR. So I think we got down to maybe we answered that. In essence, if you're doing multiple alleles per multiple targets per sample, you're down around the $20 to $40, including sequencing costs. So that's probably getting pretty close to these areas. It really depends on if you've got the PCR that you're using is an in-house assay, or if you're purchasing the assay. I think, Jenny, if you're purchasing the assay from companies that develop, especially for these repeat expansion diseases, the PCR cost alone can be a couple $100. Jenny, do you have a sense of that?
Yeah, it really depends on most people will get their kits, will get actual commercial kits, and they're of course priced more highly. Generally, if you're looking at, especially at repeat expansions, it's quite difficult to design these primers.
Yeah, fantastic. And just talking about the general tool, these questions maybe came in a little bit out of sequence, but Jenny, if somebody's going to design a new target, do we always need 2 guides per for each target?
That's an excellent question. So you might be, the person who asked this might be familiar with our previous iteration of the method. So we have had several iterations of the method to get to this more streamlined one. For this version of the method, you do need 2 guide RNAs to actually pull out the region. So 1 on each end. Yeah,
and I think if everybody's been following the gene editing area, I mean, CRISPR Cas9 and offshoots of that are incredibly efficient, as you showed an example of some guide RNAs. And typically, when we've been asked by a customer or working in collaboration with some of the early access customers, they design maybe a couple of guides on each end, and one of those is usually sufficient to move forward with the target enrichment approach. Is that reasonable, Jenny?
Yeah. So generally, for each guide RNA, at least internally, we pick 2 the design tools that we offer. Again, we have the reference guide, as Paul mentioned, I mentioned earlier. It goes through every step of this designing process, and we usually recommend picking 3 guides for each of the locus that you want to design it to, and then just pick the one that works the best. And we have had very good results. As you saw, there's almost no background or off target at all.
Yeah, fantastic. And I think in general, we're very interested in providing this capability to you, our customer base, and those who are interested in this. And then we really want to have feedback from you to us about what areas of interest in science did this new capability allow you to approach. As we've been seeing, what size ranges you're able to use and be able to sequence, and how efficient these tools are. We're very confident in the tools and as providing them, but we do really want to have this feedback from you to us. Let's see. There's a question about do we plan on developing a CRISPR RNA library? And I would say it's kind of on the same line. So the second part of the question is how to guarantee the editing efficiency and off-target effects. At the current capabilities, for the guides that we've validated, the sequence is there, and we feel confident in those. Going forward, that's one of the things we can look at is continuing to develop validated guides and maybe create a panel, but that's something that's not on our current view right now. This is the CRISPR-Cas9 is such a broad and enabling capability. We really want to get feedback from you as where we should go. And overall, as we've mentioned, the overall success of designing guides and for them working well is pretty high. So we're not concerned about creating a library or anything of that nature. Let's see. Any other questions that?
There is a question about, have you also tried shorter fragments, for example, 2 to 3 kb? So this is a good question because this might not have been obvious from the presentation. So even though we focus on 4 to 6 kb insert sizes, that doesn't mean you can't capture the shorter repeat expansions that are under 1 kb. It just means that you have more of the buffer sequence. So the insert site is just more about what we see and gets the most optimal loading on our instruments, and it does tend to like the larger fragments. So it's just adding more buffer sequence onto it, but you can go to as small of a region as you need to. You just have to add the buffer sequence to it.
Yeah. There's a question about the availability of the Sequel II protocol. That should be coming very shortly. The upfront aspects, all the way to just binding the Sequel II polymerase instead of the Sequel polymerase, are pretty much identical. Based on, Jenny showed some nice results. She's starting off with 2 micrograms per sample. So we anticipate this to be coming very shortly. If you're interested, please reach out to us and we'll provide you some detailed information. Let's see. There's some questions about service providers and enabling service providers if to be able to send samples to. Jenny, would you like to discuss that?
Sure. We're working with service providers, one in each of the geographical regions. So we have one in Asia Pacific, one in Europe, and one in US. And these are coming shortly, going to be online. So we will definitely funnel anyone through that. Now, if you are interested and you don't have access to an instrument, please email that address that or go to this, actually go to the website, and then there is a form you can fill out. So it's pacb.com/contact-us. We will then funnel you out to service providers that are currently running the assay.
Fantastic. Thanks. Let's see. I think we've handled most of the generic, most of the questions that are kind of coming in bulk. If we haven't answered your question, please, we'll reach out to you. Jenny, are you seeing any other additional questions that are worth?
Maybe one that I can easily specify. So there is about multiplexing. With 1 microgram of DNA, can I analyze multiple sites, or shall I use 1 microgram for each target? So this is actually per sample. So you can analyze several different targets with the same microgram. It's just per sample you calculate that 1 microgram. But of course, then you have to think about if you the total amount of on target calls that you're going to get is then going to be split up. But yeah, you're really looking at per sample.
Okay. I think that's the bulk of the questions. There are some specific questions which we'll get back to the individuals as well. At this point, Donald, would you like to wrap things up?
Sure. Thank you, Paul, and thank you, Jenny, for your time today and the terrific presentations you've shared. In closing, I did want to briefly mention 3 things. First, as a reminder, we have recorded this webinar and we'll have it available for download in the next several days, so please keep an eye out for a follow-up email with a link to the recording. Second, if you are interested or if you have an interesting research project and that could benefit from smart sequencing, consider applying for one of our SMART grants to win free sequencing. And for more information, please visit pacb.com/smartgrant. That's pacb.com/SMRT grant. So pacb.com/smartgrant. And please note that our 2019 Neuroscience Smart Grant program is currently open and we are accepting submissions through Friday, October 4th. And finally, to stay up to date on upcoming webinars as well as conferences and trade shows you can meet us at, please visit and bookmark pacb.com/events. So thanks again for joining us today. I hope you'll join us again on a future webinar. And with that, take care and have a wonderful day. Bye-bye.